Hello, hello, hello. Now I am audible. Can I start? I am audible. Yes or no? Can I start? Okay. Hello everyone. Welcome back to the SaraBioinformatics YouTube channel. I am Sarita. Thank you for joining today's live session. Today's we will learn the complete RNA-seq data analysis workflow step by step. Even if you are a beginner, don't worry. I will explain everything in simple language. By the end of the session, you will understand how RNA-seq data is analyzed from raw sequencing file to biological interpretation. Let's start with the basic RNA-seq stand for RNA sequencing. It is a next generation sequencing technology used to measure gene expression. It tells us which genes are active and how much they are expressed in cell or tissue. For example, if we compare healthy and cancer sample, RNA-seq help us identify which genes are upregulated or downregulated. This biomarker, hello, this technology is widely used in cancer research, biomarker discovery, drug development, and precision medicine. Here is the complete workflow. Here, here is a complete workflow. First, we collect biological sample and isolate RNA. Then RNA is converted into a sequencing library and sequenced on an NGS platform. The sequencer generates FastQ files. We check the quality of these files, trim low quality reads, align the reads to reference genome, count read for each gene, platform differential gene expression analysis, identify biological pathways, and finally visualize and interpret the result. Today we will understand each of these steps. Before starting any analysis, we need a good experimental design. Suppose we have 3 healthy samples and 3 cancer samples. These are called biological replicates. Replicates increase the reliability of the result. We also prepare metadata containing sample name, condition, and other information. Good experimental design is the Good experimental design is the foundation of a successful RNA-seq study. After sequencing, we receive FastQ files. A FastQ file contains 4 lines for every sequencing read. The read identifier, the A FastQ file containing 4 lines for every sequencing read, the read identifier, the DNA sequence, a separator, and the quality score. Most RNA-seq experiments use pair-end sequencing, so each sample usually has 2 files underscore R1 and underscore R2. The first bioinformatics step is quality control. Using FastQC, FastQC check read quality, GC content, sequence duplication, and adapter contamination. If the quality is poor, we clean the read before moving the next step. Read trimming removes adapter, sequence, and low quality bases. Popular tools include FastP, Trimomatic, and TrimGlow, Cut Adapt. After trimming, we run FastQC again to confirm that the quality has improved or not. Now we align the clean reads to the reference genome using tools such as HiSat2 our star. The output is generated in SAM, means sequence alignment map. Our BAM is a binary alignment map format. We also check the mapping percentage. A high mapping rate indicates good quality sequence data. And next we process the BAM file using SAM tools. We sort the reads, create an index, and generate alignment statistics. These steps make the data ready for gene counting. In this step, we count how many reads map to each gene using feature counts, our ST6. The result is a count matrix where row represents gene and column represents sample. This matrix is the input for differential gene expression analysis. Now we use DSEC2, our HR in R. These tools normalize the data and compare gene expression between groups. The output identifies significantly upregulated and downregulated genes based on fold changes and adjoint p-value. To better understand the result, we create different plots, a volcano plot, so significant genes, a heat map display expression pattern, and a PCA plot show how sample cluster together. These visualization make the result easier to interpret. Finding differential gene expression is not enough. We also need to understand their biological function. We use gene ontology, cake pathways, and rectome database to identify important biological process and signaling pathways associated with these genes. Next, we use the string database to study protein-protein interaction. This helps us identify hub gene and understand how protein work together in biological pathways. After bioinformatic analysis, important genes should be validated experimentally. Common methods include qPCR and Western blotting. Researchers also compare their findings with public databases like GeoTCGA. So the complete RNA-seq pipeline is FastQ file, FastQC trimming, alignment, BAM processing, feature counting, dissect analysis, pathways analysis, and finally biological interpretation. This is the standard workflow used in many research laboratories. Some commonly used tools are FastQ, FastP, Trimomatic, HiSet to star, SAM tool, feature counts, STSIG, DSIG to AdJar, cluster for filler, and R learning. These tools will help you perform RNA-seq data analysis independently. Many beginners make common mistakes such as skipping quality control, using the wrong reference genome, having too few biological replicates, or incorrect sample metadata. Avoiding these mistakes improve the quality of your result. RNA-seq analysis is a highly demanded skills. It can help you informatician, research associate, project associate, computational biologist, or clinical genomics scientist. These skills are also valuable for PhD admission and research fellowship. That brings to the end of today's session. I hope this overview helped you understand RNA-seq data analysis workflow. If you enjoy this session, please like and subscribe to SaraBioinformatics YouTube channel and share it with your friends. If you have any question, please ask them in the live chat. Thank you for joining and I will see you in the next video. Yeah, you can ask me. Please write in the comment box. Hello, I'm audible. Hello, hello, I'm audible. Okay, that's a very good question. DSEC work on count data, but it is primarily designed for gene level differential expression analysis. If your input is gene count from tools like feature count, our STSEC, then DSEC 2 is one of correct choice. However, if you want to study isoform level, our transcript level expression, you first need a transcript quantification tool such as Salmon, Callisto, or RSIM. These tools estimate transcript our isoform abundance instead of gene count. After transcript quantification, you can perform differential transcript expression using packages such as TXCMPORT together with DSEC2. Our use sleuth for Callisto output. So the workflow gene level analysis. If gene level analysis use feature count, then STSEC and DSEC2. If transcript isoform level analysis use Salmon, Callisto, RSIM. So the short answer is the yes, DSEC2 can be used after transcript quantification, but DSEC2 itself does not estimate isoform expression. It, it analyzes the count that you provide. DSEC2 is mainly designed for differential gene expression analysis. For isoform level analysis, transcript abundance should first be estimated using Salmon, Callisto, RSIM, and then imported into DSEC2 using TXIM, TXIM port. Is differential testing is required. Hello. If you have any question, you can text me in mail ID in my mail ID. Actually, this is my first live session, so if I make any small mistake, please. Sorry, I am little nervous, but I am really happy to be here with all of you. And thank you so much for joining today's live session. Feel free to ask your question in the live chat, and I will do my best to answer them. If you don't know the answer to question, I will be honest about it, and we can learn it together. Let's start today's session on RNA-seq analysis. I hope you enjoy it and find it useful. And if you have any question, please mail me Sarita Maurya 822 @gmail.com. Thank you so much. Any other question? Can I leave? Hello. If you have any question after this live session, don't worry. You can write your questions in the comment sections of this video or email me anytime saritamourya822@gmail.com. I read your questions regularly and I will do my best to reply. If many people ask the same question, I will make a dedicated video over another live session to explain it in details. If you found this session helpful, please like and subscribe. Thank you.